The important thing about amino acids to remember is that amino acids are the only molecules that contain nitrogen, and because that they do, when you metabolize nitrogen, invariably at some point it becomes ammonia. Okay? And there's got to be a specific way to get rid of ammonia in a non-toxic way, and that's why amino acid involves a unique cycle, so to speak, the urea cycle, which really is designed to take care of any ammonia produced from catabolism of amino acids. Okay, so that's very important. We'll get to that hopefully a little later today. But also importantly is that there is not a mechanism to store excess amino acids like there is to store glucose. So if you have extra glucose, you store it as glycogen. Extra fatty acids, you store them as tags. There is not a mechanism for amino acid storage in the body. We use amino acid to synthesize cellular proteins or specialized products. Any excess amino acids will be catabolized, broken down to give you a carbon skeleton, and that carbon skeleton can be further catabolized in any of the pathways we have already covered. Okay? So, important to realize that cellular proteins, you know, we are constantly breaking down cellular proteins and replacing them. Okay? Very few proteins, if any, last you a lifetime. Okay? The longest half-life of a known proteins is probably in the 15-year. Some proteins last a long time, but very few proteins last more than a few minutes or a few days. So what that means is that we are constantly replacing cellular proteins, enzymes, receptors, all kinds of specialized structural proteins. And that process of making and breaking old proteins, replacing them with new ones, is called protein turnover. Okay? So that number is somewhere around this number, 300 to 400 grams per day. So we break down about 300 grams, we resynthesize about the same amount to replace what was broken. So here you can see that most of what was broken down is going to be replaced to make new proteins. But we also have important dietary input. That dietary input will be used in 3 different ways. One, to help with the resynthesis of body proteins, cellular proteins. Synthesis of specialized products, which we'll get into towards the last lecture this term, which is some of these important products like nucleotides, like neurotransmitters, like heme, and so on. These are nitrogen-containing products that are derived from amino acids. The third fate, if you want, of amino acids can be to generate energy, use the carbon skeletons. Once you remove the nitrogen, you get carbon skeletons, and use those carbon skeletons either for energy to form glucose or ketone bodies, or degrade them through all the way to CO2 through the TCA cycle. Okay, so the middle portion here is energy, specialized products. The bulk of really the amino acids are going to be to replace cellular proteins. And that's really the same thing. It's an older picture, simpler in a way, but one of the important things about what makes a protein more likely to be broken down. For example, certain enzymes are broken down pretty quickly. They're no longer needed, so they're degraded pretty quickly. So there are specific and not so specific signals that really result in protein degradation. The first non-specific signal is here, oxidation. You know, if a protein is damaged by oxidation or other modifications, there are cellular mechanisms to kind of get rid of it and remove it from the cell. However, there are two more specific mechanisms that destine protein for degradation, faster degradation, if you want. The first one has to do with the primary sequence of a specific polypeptide. So polypeptides that are rich in specific amino acids, or abbreviated PEST sequences, and these PEST sequences are proline, glutamate, serine, threonine. So certain polypeptides that are rich in these sequences are more likely to get degraded than other polypeptides. So that's a function of the primary structure of that polypeptide. The third important degradative pathway of proteins is really very specific and very important. It's called ubiquitination. It's basically tagging a protein, flagging it, so to speak, so it will be destined for degradation. Ubiquitin is a small protein itself. Once the cell no longer needs an enzyme, there's a process that attaches this ubiquitin molecule, or many of them, to that enzyme, let's say. Once that ubiquitin is attached, it destines that protein to go to an organelle called the proteosome, where it will be degraded by proteases inside the proteosome. So this is kind of a quick representation. So here's a protein, this is ubiquitin. You can add one or more ubiquitin molecule. That labels that protein as we no longer need it. So it is sent to this organelle called proteosome, where it is broken down into peptides. Many enzymes are gotten rid of. Some other cellular proteins no longer needed. So this is a very specific way for protein degradation called ubiquitination, or adding this tag of ubiquitin for protein degradation. Okay, you don't need to know the specific details here, but just know that when you add ubiquitin, you send it to the proteasome and you degrade the protein. So as I mentioned before, really the main function of dietary protein and or amino acids that are turned over from cellular proteins is to remake cellular proteins. And that's a lot of proteins in the body. Again, enzymes, receptors, structural proteins, specialized storage proteins, specialized carrier proteins, and so on, serum proteins. So that's what we use amino acids from the diet primarily for. The secondary source is their use as energy. If you like this channel, please subscribe. Thank you.